Okay, what's on the socks? Are those drinks?
Yes.
It's nice. It's very nice. It's very nice. Brooks. Brooks. You can talk. Y'all, please make sure your masks are covering your mouths and noses. Y'all, everybody mask. Mask up. Okay, please make sure you put your name on the seating chart. Please make sure your name is on the seating chart. And make sure you check in on iClicker. iClicker's running. Make sure you check in. So iClicker and the seating chart. All right. So, I gotta remember my joke for today. What kind of a haircut do you give a bee? A buzz cut, right? Why is a bee's hair always sticky? Because he uses a honeycomb. All right. They were cute, right? Full screen. All right. So this is what we were talking about last time, right? Protein staining after separation. So if I want my protein of interest, what do I do from the beginning? What do I do? I need to first get a whole cell homogenate by breaking open the cells. Once I break open the cells, what do I do? Differential centrifugation, right? So different speeds and different times give me different fractions depending on what I want. Okay, now I've collected the fraction that I want. What do I do with it? I salt it out at a particular salt concentration. So I do a salt concentration that's lower than that of my protein of interest, right? So that other things precipitate out. And then I keep just the stuff that's still soluble. Then I add more salt to make my protein of interest precipitate. And then what do? Well, I have to spin it first. So I get a pellet. And then I resuspend that pellet and I dialyze that new sample. Okay, so dialyzing does what? Takes the salt out. So now I have my protein of interest at the correct pH, at the correct salt concentration, in the correct buffer, basically. And what do I do with it? I do some kind of column chromatography. You could do them sequentially, all three. You could just do one. It depends on a lot of different factors. But what are my three kinds of chromatography? Gel chromatography, which separates based on size. Then ion exchange, which separates based on charge. And then affinity, which separates based on binding affinities, right? So depending on which column you use, sometimes things elute out on their own, and sometimes you have to competitively make them elute, right? Okay, so I collect my fractions, and I want to check them for what? Purity, but like there's a term, like after I get all my different steps, I've collected a sample at each step, and I want to check two things at each step. Each step, check total protein and what? Protein activity. And then what do I do with this information? I take the activity divided by total protein, and I get specific activity. Right? I do that for every step. And then what do I also do? At every step, I also check it on a gel. Right? Yes, ma'am. Each step of the purification process. So after each centrifugation step, after you salt out, after you dialyze, after, like at each step, you take a little aliquot out and you check it. Because what happens if your purification scheme worked up until the last step, and the last step you lost your protein? Don't you want to know where you went wrong? So if you check it at each step along your path, you're going to figure out what happened. Okay. All right. So protein, running a gel of a protein, running a page gel of a protein, is it a purification method? No, it's a check. It's really important that you make that distinction, because once that protein's in the gel, it's in the gel. Okay. So you're not going to get it back out. It's not like you're only putting a little bit in the gel anyway, so it's not really going to help you, right? So we want to check it. So what we do is we add this dye to our protein. So our protein sample at each step, we add a little bit of dye, and then we add it to the gel and we run the gel, right? And everything runs based on charge. So what did I add to my sample? I had my sample. I added SDS, right? What does SDS do? Coats it in negative charges. So then I now have a negatively charged sample, negatively charged. And so what is it really going to run based on? Size, molecular weight, size. So this is for an SDS page gel. And then I'm going to add my dye to all of this, and then I run this whole thing on the gel. Okay. So all the dye interacts with the protein. The dye itself is negatively charged, and you're going to have some positively charged residues in your protein. And so it's that positive negative interaction that makes the dye stick to the protein, because all proteins have at least one. I mean, you're going to be very hard pressed to find one that doesn't have a single positively charged residue. And so that's just how the dye sticks to the protein. Okay, so that's how we check our sample for purification. Now, sometimes you have your protein, right? And I want to be able to characterize some protein. I have this purified protein. I was successful. Yay, it worked. Like, you should really celebrate that. Go out for drinks. Really celebrate your success as a scientist if you successfully purify your protein. Y'all have no idea how exciting that is. It really is. So let's just say I have purified my protein of interest. I was successful. Now I want to start characterizing that protein, right? What are some other properties about that protein? Well, one of the things I can do is I can do isoelectric focusing. So do you remember when we talked about the isoelectric point of an amino acid? Remember that? And it's all dependent upon what? The charged groups or the pKa's of the N terminus, the C terminus, and all of the R groups, right? Because some R groups are going to be able to be protonated and some are not. The ones that are protonated, their pKa's tell you if they're going to be protonated or not. And so each individual amino acid has a unique isoelectric focusing point, right? Okay. So if we put a bunch of protein, we put a bunch of amino acids together in a protein, and each amino acid has a particular isoelectric focusing point, can we determine the isoelectric focusing point of the protein? Absolutely, right? So the way this works is you have another gel. It's very similar to an SDS page gel, right? It has the polyacrylamide in it. But in this case, what we have is a pH gradient. What's that? Oh, thank you. Yes. So we have a pH gradient here, right? So if I apply an electrical charge to this gel, proteins that are charged are going to migrate to the opposite anode or cathode, right? So if I have a positively charged particle, it's going to go towards the cathode because that's negative, right? If you have something that is negative, it's going to go to the anode, which is positive, because opposite charges attract, right? But what happens as we go through the gel is that at different pHs, what's going to happen to those side chains and those N terminus and C terminus? They're going to protonate and deprotonate, right? Depending on what pH we're at. So when that deprotonation or protonation event occurs and the molecule is now at its isoelectric focusing point, it's going to have what charge? It's going to be neutral. It's going to be neutral. So if the molecule is now neutral, this molecule is neutral at 5.5. So it stopped migrating at pH 5.5. That tells me that the isoelectric focusing point of protein A is 5.5. This one stopped migrating at 9.4. So that tells me at pH 9.4, this molecule is electrically neutral. So let's just say I can't separate based on size. I can separate based on isoelectric focusing point. Or I've purified my protein. I want to check what is the isoelectric focusing point of my protein. I can check it on here. Right? So it's another way to check things. It's not a purification method. This is another check. It's another way to find information out about your protein. Now, this is super cool. So you can use what's called 2D gel electrophoresis. So before we explain what it is, I want to talk about a situation, right? Let's just say I have sample 1, right? And sample 1 are cancer cells, right? These are cancer cells that are untreated. Every single cell has a unique protein profile, a unique proteome, right? We talked about that. As a medical related to anything, what do you want to do to cancer cells? We want to kill them, right? We want to get rid of them. So we want to try different medical treatments to kill the cancer cells, right? So a lot of times what we want to do is have a treated sample. So we have those same cancer cells, but we give them a drug. And I want to know, does this affect my cancer cells? So this is sample 2, right? How is it affecting these cancer cells, right? Maybe it's killing them, maybe it's not, maybe it's inhibiting their growth, right? Whatever it is. But if I can understand at the protein level what's happening at the cell, like, am I down regulating this protein? Am I up regulating that protein? Then I can start to understand the biochemical reason why treating these cells with drug X works or doesn't work. Okay, so we're going to use this 2D gel electrophoresis to answer that question. So we're skipping out of, we're no longer purifying proteins. Now we're trying to do some biochemical assays. So in a 2D gel electrophoresis, we separate with 2 properties. We separate with isoelectric focusing point, and we separate on molecular mass, right? So we combine isoelectric focusing with SDS page. And you can separate the thousands of proteins that you find in a given cell individually on a gel. So let me show you how it works. You take one of your samples. For example, this is sample 1. And you put it onto an isoelectric focusing gel. So we first separate proteins based on their isoelectric focusing point. Ones that have low PIs are down here. Ones that have high PIs are down here. We cut this lane out of the gel and put the whole thing at the top of an SDS page gel. The samples are already dyed, and they're already separated based on their isoelectric focusing point. Then we run the SDS page gel. And so when we run the SDS page gel, we're going to run that based on size, right? So what are we going to have down here? We're going to have things that are low molecular weight, things up here that have high molecular weight, right? And we've already separated in this dimension based on isoelectric focusing. So you see how it's 2-dimensional? So when I'm done, this is my end product. I can look at things that have high mass and high isoelectric focusing points. I can look at things that have low isoelectric focusing points and low mass, or something in between, anywhere. But I don't just do this for one sample, right? This is sample 1. This is my untreated. But what else do I need to do? I need to do the exact same setup, but I need to do it for sample 2. This is my treated cells. I do the same thing. I take my samples, I put them onto the isoelectric focusing. So we'll separate by PI. Then we'll take those and we'll put them onto our SDS page gel. So we'll do SDS page, and we'll get another 2D gel. So we have the 2D gel for sample 1, and then we have the 2D gel for sample 2. Now, what am I looking for when I compare these two? Do you think that they're going to be perfect matches to one another? No, I'm gonna look and I'm gonna say, okay, this dot right here should be here, but it's missing. I've lost expression of a protein. Or this tiny, tiny dot here is like massive over on this gel. So what does that tell me? It's expressed more. The cell is going to react to a drug. We need to understand how the cell is reacting. And this is one way to understand how the cell is reacting in terms of protein products. So what we can do is we can take those two and we can differentially stain them so that we can overlap them, right? It's really hard to look at a bunch of gray dots and a bunch of gray dots. That's really hard. So what we end up doing is use dyes to distinguish the two samples, right? So we covalently label our samples here in the very beginning. So we label this one, I don't remember, it's blue and green, I think. Okay, so the green we'll say is untreated, labeled with green dye. And this one, sample 2, is going to be labeled with red dye. Right? So all of these dots are going to be red, right? All of these protein dots are going to be red, whereas all of these are going to be green, right? Every single one of them, I'm not going to color them all, but every single one of them are green, right? So now what we can do is we can overlay the two onto one image. Like you can scan it with the computer and have two images and overlay them. So now we should get different colored spots based on that overlay. So if you end up with a red spot, let me not circle it in green because that's so confusing. So if we have a red spot, that's telling me that, well, if it's red, red was treated or untreated. Red is treated. That's telling me that I only see that protein when I treat with the drug. If I have something that's green, green is what? Untreated. So I only see that protein in the untreated sample. So if I treat with the drug, it shuts down production of this particular protein, right? And then if I have a yellow spot is the combination, right? So then yellow protein is in both, which means it's red and green. This is called a 2D gel. 2D gel. 2D differential in gel electrophoresis. They call it, sometimes they call it a digi gel. But it's running two individual 2D gels and then overlapping them. It's really cool. It's really, really cool. So they actually show you, here's a real example. So this is the computer image, right? You see mostly yellow. Most of the dots are yellow. The intensity of the yellow varies, but it's still basically yellow. And what you can do is you can go in the computer program and you can analyze it and give it false colors. And so that's what they did for this second image. In the false colors, the green means there's no change. The blue means we increased this protein, and the red means we decreased that protein. They just falsely colored it. That's all it is. But it's a really interesting way to see what's changing in the whole cell. Now, the cool thing is I can then cut out a piece of the cell, and I can figure it's a piece of the cell. I can cut out a piece of the gel. I can say, okay, this is an unknown protein. I don't know what this protein is. So I can actually cut out a portion of the gel and I can sequence the protein. I know y'all have heard about sequencing DNA, right? Well, you can also sequence the protein. So this was begun a long time ago in the 50s by Frederick Sanger. He was the big one who started. And most people know about Frederick Sanger for Sanger sequencing. You heard that before? Most people know him for DNA sequencing, but he actually started in protein sequencing. And he designed the first method to actually cleave peptides and make them individual amino acids. But it was really, really expensive. It was really, really not cost effective. And so he switched to DNA. And so he got another Nobel Prize for that sequencing. So he has gotten 2 Nobel Prizes, one for sequencing proteins and then one for sequencing DNA. Very, very cool individual. So I told you that the Sanger method of protein analysis was kind of expensive, didn't work so well, but it worked. But there was an improvement on it, and that improvement is called Edmond degradation, and this is what we use today. And so you take basically a chemical called phenol isothiocyanate. We call it PITC for short, and we covalently link it to the N-terminus. And then we treat it with an acid that will cleave only the PITC and the one amino acid that's attached to it. And so we call this is trifluoroacetic acid. And so this acid will cleave the PITC that's linked to an amino acid. And so you can cleave them off one at a time. So the peptide's always cleaved between the first amino acid. The first amino acid, it's really PITC plus the first amino acid. So this really comes off. And then the second amino acid is attached to the rest of the protein. So you can take them off one at a time. It's really a really interesting process. And you can program a machine to do it so it can become automated. And you can do this with a peptide that's as long as 50 amino acids long. Are proteins longer or shorter than 50 amino acids? Typically longer. So we're going to have to do something to our proteins before we can do this. Let's say, so say I have this protein. I have protein X. I isolated it from a digigel, right? Okay. I want to know its sequence. What do I do? It's really big. Can I go right into sequencing? No, I can't. I need something that's going to chew up my protein. What does that in the cell? Proteases. So I'm going to treat the proteins. I'm going to do protein plus some kind of protease. And from that, I'm going to get oligo, O-L-I-G, oligopeptides. Those oligopeptides are going to be less than 50 amino acids long. Now that I have oligopeptides, what can I do with them? I can do Edmond degradation, which is sequencing. And I can get the amino acid sequence. So you have to think about what are the limitations of each of the techniques and what they can and can't do to know what steps you have to take. So this is how Edmond degradation works. In Edmond degradation, we have this polypeptide chain, right? Here's in red is residue 1, 2, 3, right? So when you treat with PIT-C, what happens is this carbon right here of the PIT-C is going to create a covalent bond with this nitrogen right here. When I treat this with a base, what's actually going to end up happening is that you're going to have the electrons that are in this sulfur carbon double bond here come and attack this carbon. So you're going to break this bond right here. What is this bond right here between the C and the N? That's the peptide bond. That's exactly right. This right here is the peptide bond. It's too far away to attack the next peptide bond. It has to be the closest peptide bond. So if we then cleave it, we add trifluoroacetic acid, we cleave it, and it creates basically a ring structure, and we can get this one amino acid residue attached to PIT-C. So we call this the PTH amino acid. So this one we can put through, for example, chromatography methods. You can do mass spec, but you can identify which amino acid is hooked to the PIT-C. And so from this, we can do one at a time, as long as it's no longer than 50 amino acids, and we can identify our protein. That's awesome. If you know your amino acid sequence, can you go find it in the genome? Yeah, I can translate protein sequence back to DNA sequence. Then I can find out exactly where the gene is that's controlling that protein. Well, that's going to give me a lot of information, right? So you just got to think about all the different ways that this can be used. I mean, there's biochemistry has come so far in such a short amount of time. So remember we said you can only do proteins that are 50 amino acids or shorter. So if you have one that's longer, you have to cut it up with proteases. Some examples of proteases are trypsin and chemotrypsin. Proteases. So the deal with proteases, and we're actually going to study some of these enzymes in detail in, I think, the very next chapter, is that they're very particular in where they cut on the protein. Trypsin will only cleave on the C-terminal side of arginine and lysine residues. So these proteases are enzymes, right? What does an enzyme have to have? A binding site, an active site, right? Where it's actually going to undergo its catalytic activity. That's the definition of an enzyme. So the pocket of actually where that enzymatic activity happens, what do you think those residues, what do you think the charges are in those residues? If we're interacting with lysine and arginine, what are lysine and arginine? They're what? They're positive. So then what's going to be in the pocket? Negative residues. Okay, so think about that. Why is it cleaving on both lysine and arginine? Because they are the same charge. Chemotrypsin cleaves on the C-terminal side of, what is TYR? Tyrosine, TRP, tryptophan, PHE, phenylalanine. What do these three have in common? They're aromatic rings. So this isn't necessarily a charge, but it's a size and a conformation inside that enzymatic pocket that's going to allow those huge aromatic residues to fit. So enzymes are very, very specific for their substrates. Okay, there are some other ones. Cyanogen bromide is on the C-terminal of methionine. What does methionine have? Sulfur groups. Staphylococcus aureus has an enzyme that we typically use that cleaves on the C-terminal side of, what is ASP? Aspartate and glutamate or glutamine? Glutamate. Okay. So regardless of whichever one you do, you're going to have a really long protein. So here's your protein. It's going to be really long, right? What do we do to that long protein? We're going to cut it with two different proteases. So we're going to take that long protein and we're going to do one thing to it is to cleave it with trypsin. The second thing we're going to do it is to take a little bit of that same sample, cleave it with chemotrypsin. So I take these samples, right? I have these little fragments. Are these less than 50 amino acids long? Absolutely, right? They're much smaller. So I'm going to take those and I'm going to figure out their exact sequence by what? They don't say it in this diagram. I'm going to take them both and I'm going to do what with them? Either Edmond degradation or what else could we do? Mass spec. We can mass spec them. And we can find out their particular mass and then figure out which amino acids are in that polypeptide chain just because we know their mass, because we know the mass of each amino acid. So once we get our sequence, what you do is you actually line them up. And you do this, you can do this by eye, you can do this by hand. It's not that hard. And you just overlay the parts that are the same, the parts that are overlapping. And then you read it from left to right. And that's what the bottom sequence is. And that's how you can figure out your exact protein from start to finish. Isn't that really cool? Yeah. If we have time, I have a protein for us to analyze, but I don't know that we'll have time. All right, so we talked about mass spec. Yep.
So I understand, I guess, why they're being used. You said that for those certain, I guess, proteases, for the example, if it was less than 50,
Yeah.
how did they use, I guess, those proteases that were meant to be higher than 50 on that?
What do you mean the ones that are meant to be higher than 50?
Like the proteases for the protein cleavage.
Here.
It was for proteins that were longer than 50 residues.
Right. So if you have a protein that's only 40 residues long, you could do Edmond degradation and be done and figure out your sequence, right? But if you have a peptide that's 1,000 amino acids long, there's no way you can do Edmond degradation on that. So what you have to do is chop it up with the proteases, trypsin, chemotrypsin, and then sequence those individual little polypeptides and put them all together to make your final. Does that make sense? Are you good on that? Ask me if you're not. Okay. All right. So we talked about mass spec. Mass spec is a way for us to identify which amino acids are in a short polypeptide, right? So how does that work? Well, what we want to do with mass spec is to find the mass of a short polypeptide fragment. And so really what you're actually measuring is a mass to charge ratio when you do mass spec. And so once you get that mass to charge ratio, you can convert that to a molecular mass. But how does mass spec really work? So in mass spec, basically what you do is you do an applied electrical field, right? And when you apply an electrical field to charged particles, what happens to those charged particles? You're going to exert a force on them, right? They're going to move. So you apply electrical field to charged particles. So the field, and I feel like your book should explain this a little bit better, but your field exerts a force on the molecules. Okay, and so when you apply that force, you basically charge your molecules and then you shoot them out. And if it was perfect, they'd go in a straight line, right? But when you apply a magnetic field to them, it pulls on the ionized molecules and they take more of a curved route. What's going to affect that curve? Mass, the mass. Basically, you give them all the same charge. You have to artificially ionize them, right? These are charged particles. So really, the charge is dependent upon the mass. So the more massive, they're going to curve at a different angle than a less massive, right? And so you can measure that change in path and from that determine your protein sequence. It's really cool. It's really, really, really cool. So I told you you have to do it on charged particles, right? Well, how do you make sure that all of your stuff is charged? You're going to ionize your peptides. And there are two different ways that you can do this. One is the electrospray ionization. When you ionize something, you really, really run the risk of basically destroying your protein. So this is really finicky in how you do that. And so basically what we want out of this, we want to cut it up with trypsin, right? We get little trypsin polypeptides, and then we want to have it in a gaseous phase. So you basically put them in a solvent that's very, very volatile, right? And so that you can ionize them. So basically what you want out of this is you're going to get a highly charged gaseous particle, or like polypeptide. Oligo is really not a poly, really we could say oligopeptide. Right? So now that we have it in the gaseous phase, we can shoot it through our mass spec, apply a force to it, and see how it curves. The other way that you could do it is you can use a matrix. So this is Maldi or Malditoff Depends on, what you're using with it. But you do the same thing. You trypsinize your fragments, and they're embedded on a matrix, and that matrix will absorb light. And so you shine a laser on the matrix, and then as the molecules are hit by the laser, they become charged. And when they become charged, they're released from the matrix. So now you again have a highly charged gaseous particle ready for mass spec. So either way works. All right, so what happens? This is kind of a typical mass spec diagram. What you want to do is you want to start with ionized molecules, right? So you have relatively big ones. And so what you do is you ionize them, and then you run them through the first mass spec by applying that electrical force. So you start to have some separation. Then what you do is you fragment those peptides again. So now, do you see how we have smaller and smaller pieces? And we do the same thing. We run it through a mass spec again. We're going to separate them again, and then we start to detect them. So we can't detect things that are really big. We have to wait until we get them really, really small. And so you get a spectrum out of there, and each of these peaks is going to correspond to a particular molecule. And then you can look at the mass of the peaks and predict based on the peaks. And we're not going to do that. You have to, that'd be like a whole class. You would be able to predict your amino acid sequence. That's like a whole class. Awesome, but we're not going to do that. So then what you do is, what do you do with that information? Right? If I did this in lab and I got out a protein amino acid sequence, hey, really cool, but what am I going to do with it? Am I the only person researching proteins in the world? No. There are lots of scientists who are doing it. So you put it into basically a database. And you say, okay, well, maybe I'm working on this mouse protein, but somebody did this study in monkeys, I don't know, whatever it is, or on humans. And I'm able to say, okay, this matches really, really closely with their stuff. So it must be a homologous gene, right? So even if somebody hasn't worked on the particular protein that I'm trying to identify, usually you can predict what it is. So you always do some kind of database, right? Okay. So we can analyze a protein that we already have, which is really, really interesting. But sometimes we want to make a particular protein, right? And there are different ways that you can do it. You can hijack like a bacterial cell. You can put the gene in and you can tell that bacteria, you make this protein for me, right? And you do that by changing the promoter region, right? And that kind of stuff. Or you can actually synthesize a protein from scratch. So a lot of times, it's relatively difficult to do. So typically we don't do it for large proteins. Typically we do it for smaller proteins. One example of a smaller protein that we like to synthesize are antigens. So let's just say I want to make antibodies for COVID, right? So if I know the spike protein on COVID that binds to the antigens, right? That is the antigen that binds to the antibody. I can synthesize just a really small fragment of the spike protein, and I can give it to doesn't matter, any given animal, right? Or you could give it to a cell in culture, a B cell that we pulled out of a human, and we can give it to it in culture, and we can make that B cell make the antibodies that we want it to make. And then we can grow it in the lab and amplify it, and then we can send it out and then we can actually treat patients. The other thing that we can do is we can create antibodies this way for research purposes. And I'm going to tell you all the different ways that we use antibodies for research purposes. This is the whole scheme for how you synthesize peptides. And I don't want you to memorize it. I'm not going to like ask you stepwise on the exam how it works. But I just kind of want to look at it really, really quickly. When you make proteins in the cell, how are proteins made? In what direction? N terminus to C terminus or C terminus to N terminus? N to C, right? This is different. It's the opposite of what happens actually in the cell, right? So in this process, what you're going to do is you're going to actually add to the C terminus here. So you're going to start with whatever protein it is. You're going to attach it C terminus to N terminus here. You're going to cleave off here, and you're going to add another one. So here is actually what portion of the molecule, like here and here. We have our C terminus, we have our N terminus, right? So do you see how we're adding to the N terminus? And that's supposed to be the beginning of your protein. But in order to make this work, what you have to do is you have to attach the C terminus to a resin. And so if it's attached to the resin, you can't add anything to it. So you actually end up adding on the opposite side. And then when you're all done, you just cleave the resin off, and then you have your fully made peptide. But this is really expensive. And so we're only going to really do it for really small peptides. Well, when do you only need really small peptides? When you're making antigens, right? Antibodies recognize really small things. It's not the whole spike protein that an antibody will recognize. It's only a small portion of it. So we can use it in making antibodies. And the very last section, we talk all about antibodies. Okay. So if you're doing protein science, you've purified your protein. Congratulations. Awesome. Now, you really want to know what it looks like. You can look at the sequence and predict alpha helices. You can predict beta strands, beta sheets, but you don't know what the overall three-dimensional shape is until you actually look at it. And there are two different ways to look at it. There's X-ray crystallography, and there's NMR, nuclear magnetic resonance spectroscopy. So X-ray crystallography was actually in the late 1950s that we actually first started doing this. You don't have to write it down. I'm not going to test you on it. Sometimes I forget that when I write it down, y'all are automatically going to write it down. But they give you two different kinds of information. So when we talk about X-ray crystallography, the first thing you have to do in X-ray crystallography is you have to crystallize your protein. Is a crystal a static thing, or can it move? It's static. Are proteins static in the cell? No. So if you do X-ray crystallography, you're doing what's called a snapshot. You only get one picture. Now, a lot of proteins have binding partners, right? Or they have ligands. They have different things that they bind to that add to their stability. So a lot of times when we do X-ray crystallography, we actually give the protein a little piece of their binding partner in order to stabilize the conformation. And then we do X-ray crystallography. We crystallize it with the ligand in place. So it's limited, right? You're only going to get certain kinds of information out of it. NMR, on the other hand, is not. It's not static. You actually apply a magnetic field to your atoms, and you change the spin of the electrons, of the atoms, the nuclei in your atoms. And when you change the spin, you actually excite them. And when they come back down from that excited state, you measure that difference. So does it matter what shape they're in when they do that? No. So you're going to do it like multiple times, and you get multiple images, and you kind of combine them. But there's a problem with NMR. NMR is only good for small proteins. So they each have their drawbacks and their perks. So NMR is typically for proteins that are 40 to 100 kilodaltons. If your protein's out of that range, NMR is not that good for it. It's not good really small, and it's not good really big. So they both have perks and drawbacks. So let's talk about each one. So in X-ray crystallography, we actually shoot X-rays at a molecule. And it really, the hardest part of X-ray crystallography is getting the crystals. Have you ever seen a science movie where like they're trying to treat some like contagion or, you know, something like that, and they're studying this protein and they go in the lab and they crystallize it overnight? Bull crap. Years, sometimes it takes years to get your protein to crystallize, because you don't want to make if you change the salt concentration, what could happen to your protein? It could denature, it could aggregate. Well, a three-dimensional picture of an aggregate is junk, right? So you have to crystallize in the native state, which is really, really difficult. So sometimes years in order to get, and sometimes you never can. There are some proteins that you really, really can't. Then once you have that crystal, then you can actually look at how X-rays are diffracted by the crystal. Okay, so let me show you how this works. I'm going to use this chair. So let's say that this chair is my protein of interest, right? Okay, I turn the lights off. Can you see the chair? If the lights are off, you can't see the chair. Let's go with that assumption. So basically what you do is you stand over here and I'm going to shoot a gun at it, like a machine gun, right? A bunch of bullets. But here's the deal. This chair will not get hurt by the bullets. The bullets will just bounce off. Where are they going to hit? All around it, behind it, right? At different angles. Okay, I get one picture in this orientation. This is my crystal. I'm going to take my crystal and I'm going to move it in a different orientation. And I'm going to hit it again with my machine gun and get a diffraction pattern. So that's what you're seeing here. I took the crystal, the X-ray beam is my machine gun, and I shoot the crystal. Well, the electron clouds will make that X-ray beam bounce and hit the wall, right? It's not going to absorb it. It's going to bounce and hit the wall. So when it does that, you can pinpoint where those rays are diffracted on the detector, which would be like the board. And you take that crystal, you rotate it. You get a new diffraction image. You rotate it. You get a new diffraction image. You rotate it. You get another one. You get the picture, right? We combine all these 2D diffraction images into a three-dimensional electron density map. Then, because we know the atoms that make up each individual amino acid, we can predict which residues are in which place because of the electron density. And we can make a protein model from that. Isn't that cool? It's awesome. So what I did for y'all is, because I think it's so cool, I pulled a bunch of extra little videos if you're like super interested in it. There's two different, oh, that's weird that got mixed up. I'll fix it. But there's two different X-ray crystallography videos. There's an NMR video. There's one on Edmond Degradation, one on mass spec. They're all really cool. Okay. If today's lecture was just not enough for you. All right. So how does this help us, right? What does it matter? So this is an actual X-ray diffraction pattern that came from the X-ray crystallography of this protein called nitroporin 2. Nitroporin 2 is made in insects, blood-sucking insects, right? And so what they actually found in this molecule is a heme group. They were able to find the heme group based on just this X-ray crystallography. Heme can bind to nitric oxide. Why would an insect that wants to suck your blood, a mosquito, want to bind to nitric oxide? What does nitric oxide do? It's a vasodilator. Why would a blood-sucking insect want to have a protein in its saliva, maximize blood flow, and it will get more to eat? Isn't that awesome? So these are like some of the things that just have been discovered by using X-ray crystallography, right? Same thing with DNA. The shape of DNA was discovered by X-ray crystallography, right? Okay, so the second method, NMR. NMR really looks at just a few atoms within the entire molecule, right? So we know we're mostly carbon-based. We have lots of nitrogen in proteins. We have hydrogens in proteins. So we look at hydrogen, nitrogen, and carbon, and we look at particular isotopes of those atoms. And so what we do is we apply a magnetic field, and that magnetic field aligns all of the nuclear spins of these individual atoms. But they don't stay that way. They're going to revert back to their native conformations, their native spins, and that change in energy we can measure. And so that's how we get nuclear spins. That's how we get NMR pictures back. So this gives us a really dynamic structure. Oh, I thought I had a picture. In your book, there's a really nice picture of actually the difference. And you'll see in an NMR image, you'll see more of a fuzzy line, right? And where maybe alpha helices are or where beta sheets are. And so you can actually see the different portions of the molecule that are more flexible than others, right? The whole protein is usually not super flexible. Usually there are certain areas of the protein that are flexible, and NMR will tell you that. So it's very powerful, but it's very limited in size. You cannot go over 100 kilodaltons, and usually under 40, you don't get that much. Your data is not so great. So a lot of times what researchers actually do is they do both crystallography and NMR. And then you combine that data and you make a molecular map of your protein. So why is that important? Then you can model in mutations. So I know this residue being changed to a positive to a negative, right, is in the enzymatic pocket. And so that's why this person has this particular disorder. It gives you lots of information. Okay, questions on that? Good. Interesting. All right. So last thing we're going to talk about are protein-specific antibodies, but I think we are out of time. So I guess we will finish this one. It's not very long. We'll finish this one next time. So when's the homework due?
Tomorrow night.
Okay, so I'm going to extend it to Sunday night because our next class is Monday. All right, any other questions? All right, anything on Zoom? Isn't today really interesting? I say that on every class though. All right, so I'll go ahead and end.